Good afternoon, everyone, and welcome to the fourth session of the National Beef Cattle Evaluation Consortium's Brown Bagger for 2019. For those of you expecting to hear Bob Weber's voice, this is not Bob Weber's voice. I'm your host today. My name's Darrell Bullock. I'm an extension professor at the University of Kentucky, and I would like to thank my partners, though, Dr. Weber at Kansas State and Dr. Matt Spangler at University of Nebraska. The three of us are the ones that coordinate the educational program for the consortium, and we would all like to welcome you to the last session of this year's event. As always, I'd like to remind you guys that that we do have a website, and it is still maintained. Primarily, the information going on there at this time are the recordings from these sessions, and we have those archives from years past. And shortly after today, as soon as Dr. Weber can get everything organized and worked out, we will get them posted again this year. So if you don't see those, give us a shout, and we'll make sure and get them put up. But it shouldn't take too long. We'll have all the recordings after today and get those put up for this year. Today, we're welcoming two folks, actually from the University of Florida, both of them today, to talk about completely different topics. But our first topic today is going to be genetics of meat quality in Bos indicus cattle. And this is going to be presented by Dr. Raluca Mateescu. And Raluca actually got her bachelor's degree at Bucharest University in Romania, and then she got her master's and PhD in animal breeding and genetics from Cornell University. Raluca's first position out was at Oklahoma State University, where she spent 7 years. And then in 2014, she moved to the University of Florida, where she is a professor of quantitative genetics and genomics in the Department of Animal Science there at the University of Florida. Raluca's work primarily focuses on genetic markers associated with economically important traits, and as many of you know, Raluca does a lot of work with Bos indicus cattle, which is a little bit maybe of a missing spot in a lot of the research, and she's kind of filled that void and does a really good job of keeping us up to date on how genomics is working within the Bos indicus species as well. So Raluca, I welcome you today, and I am going to stop sharing and turn it over to you, and you can take control, and look forward to hearing your talk today.
Good. I trust that you see my screen.
Not yet. You got to hit the share button, I think, is what you're probably missing.
Oh, we did practice, and now I messed it up.
I think once you've identified which of your screens you're going to do, there's still a button down at the bottom where if you hit, it'll show share. There, it's coming. That's it.
All right, very good. Thank you. And let's see, I messed up my... All right, very good. Well, thank you very much, Dar, and thank you for the invitation. Really excited to be part of this year's NBCEC Brown Bagger series, and very excited to be able to share some of our work on genetics of meat quality in Bos indicus influenced cattle. So I'm going to start by arguing that beef product quality and actually meat quality is and should be a top priority for the beef industry, and that's mainly for two reasons. First of all, it has a great power to influence demand, but also because the industry has the ability to improve it. And this is even more important when we look at Bos indicus crosses, because they are routinely penalized for relatively low marbling score, but also probably even more important because they are routinely penalized for this perceived inadequate tenderness. And I'm going to show you some data related to tenderness in just a second. But first of all, let's just look at meat quality and how do we assess meat quality right now. So what we have in the industry, we use the USDA grading system, which everybody knows is based on marbling and maturity. And it does a great job at assessing marbling, which is what was supposed to do, but it has a limited power in predicting eating quality. So when we look at, or when we talk about eating quality, I'm really referring to tenderness as the main driver, because when we ask consumers, they consider tenderness to be the most important factor that affects their eating satisfaction. And that's where the USDA grading system is really not able to assess the tenderness or the eating quality of our products. And I want to show you a little bit of data to this point. And this data is coming out of our University of Florida herd. This is a multi-breed herd, but it's really a population that has everything from 100% Angus to 100% Brahman. We group these animals in 6 groups, with breed group 1 being 100% Angus, breed group 6 being 100% Brahman. Breed group 3, you see it here, is the typical Brangus 3/8, 5/8, but it's really, again, a continuum from 100% Angus to 100% Brahman. And I had about 1,366 cattle in this population, and I wanted to look at the relationship or the lack of relationship between tenderness and the USDA quality grade. So first of all, when we look at this population and just the way it grades, pretty much like you would expect, just because it has a pretty big Brahman influence in this population, we have about 47%, almost 50% that would be select, graded select. But you do have about 30%, 32% choice minus, and about 14% that would grade choice or choice plus. Again, probably not surprising given the strong Brahman influence in this population. But what I would like to show you is actually how this population looks like when we look at tenderness. So in this case, tenderness we measured using the Warner-Bratzler Shear Force. This is a test that measures the force it takes to shear a steak, or really, in other words, it measures how much force you would need to chew a steak. We measure it in kilograms, and if we look across within each quality grade in our population, you can see the distribution in terms of Warner-Bratzler Shear Force. So the blue dots are the individual steaks, the Warner-Bratzler Shear Force for individual steaks. The orange dot would represent the average for that particular quality grade. And then I also put a line, the red horizontal line shows you the threshold, so 4.3 kilograms of Warner-Bratzler Shear Force is really the threshold that we call under 4.3, we would consider steaks tender or very tender, and then above 4.3 would be tough. Again, keeping in mind that a higher number means you need more force, means tougher steak, and the lower number is the one you're looking for, meaning you're looking at a more tender steak where you do not need a lot of force to shear through it. But really the point of this graph is to show you the amount of variation you have, and if you only look at standard and select, you can see that even within the lower quality grades, you have a lot of steaks that would qualify for either tender or very tender. Probably, half of those individuals would be tender or very tender. So although the quality grade does a great job at characterizing carcasses in the degree of marbling, it doesn't do a good job at assessing tenderness. And again, it's really not a criticism, because the quality grade, the USDA quality grade, was not designed to do that. But this is a problem that we have if we are trying to look at the tenderness of our products. I also wanted to show you the variation in Warner-Bratzler Shear Force in a different way. Again, going back to that idea that the Brahman or the Bos Indicus cattle are very tough. So I wanted to show you the variation in tenderness by breed in our population, and I'm going to use the same multi-breed population, but I'm going to show you only the purebreds. And I'm going to start with the purebred Angus population. Again, if you look at this, this is the distribution in Warner-Brasler shear force. Keeping in mind 4.3 would be your threshold, right? Below 4.3, tender or very tender. Above 4.3 would be tough. Not surprising, just like any other Angus population, you have the majority of the animals with tender or very tender, but you do have some animals, even purebred Angus, yes, I know, surprising, that would have a tough steak. So probably even more surprising is if you look, I'm going to superimpose the Brahman data. So these are purebred Brahman cattle, and we are looking at the Warner-Brasler shear force. Now, in blue, you can obviously see, yes, we do have more animals on the right side, which means a little bit tougher steaks, but there is also a lot of overlap. So, and still probably half, if not a little bit more than half of our purebred Brahman animals being tender or very tender. So I would like to make sure that people take this message and saying, you know, the purebred Brahman, we had a lot of advances in our breed, in this breed, and even now if you look directly at tenderness, it is probably a little bit different than what a lot of people would imagine to be. But I'm going to go back, okay, so if tenderness is what we are interested in, right, and it's really difficult to predict using the USDA grading system, we do know we have genomic tests. However, when we look at the genomic tests that are on the market, they have been developed mostly on Bose Torres data, and the problem with that is they do have a limited prediction power in Bose indicus influenced populations. And so what I would argue is that what we need to have genomic tests that are breed or even population specific. And I'm going to show you just one example of why am I trying to make this point. So I'm going to look at a genetic test, and I'm going to show you actually just one specific marker. This happens to be a marker in Kaltestad, and I'm not going to tell you which one it is, but it is a SNP, a single nucleotide polymorphism. As you know, that means a variation at one position in the genome. So in this case, we have either a C or a T at this particular position, which means in a population, I can have three possible genotypes, either two copies of the C allele, a heterozygous, or two copies, a homozygous for the T allele. So when we look in our, and again, I'm going to look at the purebred Angus and the purebred Brahman population. If I look at only the purebred Angus group in our University of Florida herd, you can see that this particular marker is associated with the Warner-Brasler shear force, and you can see the difference with individuals which are homozygous for C, carrying two alleles, two copies of the C allele, having more tender steak, so 3.69 versus maybe a 3.96 for individuals which are homozygous for T. So a difference of about 0.3 kilograms in Warner-Brasler shear force between these two groups of individuals, and this is only from one particular marker, which is great. The problem is that when I look at exactly the same marker, but in the purebred Brahman population, I see a different picture, and I still have an effect of this particular marker, but in this case, the beneficial allele, which was C in Angus, now a homozygous for Angus in the purebred Brahman, is actually going to be associated with the tougher steak, this group having an average of 4.73 kilograms of Warner-Brasler shear force. So if you look at the difference between the homozygous for T, homozygous for C, it's still about a 4.3 kilograms difference. However, which allele is the beneficial one? It's completely reversed. So we find this with a number of markers for tenderness in our populations, where even though those markers might have an effect, the effect or the beneficial allele is sometimes exactly the opposite ones. And so that's why our overall goal is to develop these genomic tools that would allow us to select superior meat quality in both indicus influenced populations, with the idea that the tests that we have now on the market are not working very well in these particular populations. And so I'm going to go over some of the strategies that we are using at different levels to deal with these complexities that we have in the meat quality, and how do we try to get to our goal of developing genomic tests that work in our populations. And I'm going to start with at the phenotypic level, a lot of complexities here, just because the way we define meat quality, we can use different traits, right? We can absolutely, marbling is part of meat quality, but we can also use tenderness, either measured by Warner-Brasler shear force, tenderness from a sensory panel, but also juiciness, connective tissue, beef flavor, off flavor. So a multitude of traits that all of them together would describe what we call meat quality. And so in this context, we wanted to use this structural equation modeling to deal with the complexity at the phenotypic level. The structural equation modeling allows us to derive a network of unobserved latent variables, and the latent variable is just something that I cannot measure directly, but I can derive from a multitude of observable phenotypes. And I'm going to give you an example just to show you what I mean. For example, I have a number of traits I can measure from a sensory panel: tenderness, juiciness, connective tissue, and flavor. These are observable phenotypes, but all of them are describing a latent variable which I cannot observe, which I can call meat quality. On the other hand, I have other type of traits like hot carcass weight, dressing percentage, marbling, ribeye area, and so forth. Again, I can measure those traits directly, but all of them are describing what I can define as carcass quality. So that would be the latent variable. And then I also have a number of other traits like birth weight, weaning weight, but also average daily gain from birth to weight, or weaning to yearling, and so forth, which I can measure directly, but they are all underlying an unobservable latent variable, which I can call growth. And then I also know I have different relationships between these unobserved latent variables, in a sense that I know the growth will have an effect on carcass quality, and carcass quality will also have an effect on meat quality. And so the structural equation modeling allows me not only to derive these latent variables, but also the relationships between them. And so also, another appealing part of this approach is that we can use the structural equation modeling combined with a genome-wide association study, and this is a very powerful approach to uncover different genomic regions associated with the latent variables. First of all, it will allow me to identify genomic regions which are directly controlling a latent variable. For example, this particular region could have a direct effect on meat quality. I could also identify genomic regions which have an indirect effect in latent variables. For example, here at the top, I have a genomic region which has a direct effect on growth, but because growth has an impact on carcass, this particular genomic region will also show an indirect effect on carcass quality. And then probably the most important type of regions are those with a pleiotropic effect. Those are genomic regions that would have a direct effect on two or more traits at a time, for example, this one having a direct effect on carcass quality and meat quality. So this would be what we call a pleiotropic effect. And so this strategy allows me to differentiate between these different types of genomic effects. So we did this, we used this approach in our multibred population here at the University of Florida. Again, these are animals that range from 100% Angus to 100% Brahman. We had at the time 726 steers available with 22 observed phenotypes and also 250K genotypes. And our final structural equation model included the carcass quality as an independent variable. The carcass quality was measured by fat over the ribeye, marbling, which was the strongest variable for this latent, for the carcass quality latent variable, and also quality grade. And then the meat quality was our dependent variable, and this was measured by connective tissue juiciness and tenderness. The growth actually did not fit our structural equation modeling, meaning it didn't have enough or a strong enough association with the carcass quality and the meat quality. But this allows us, first of all, to derive these two latent variables that now we can use to look at genome-wide level, but also allows me to look at the relationship between the two and try to model that relationship. So I'm going to move now to the DNA level. So I took the latent variables that we derived at the phenotypic level, and I'm going to go to the DNA level where we are going to look for those genomic regions. Like I said, we can detect either genomic regions with direct effect on carcass and meat quality, but also genomic regions that have a pleiotropic effect or indirect effect. And when we did this, we identified about 571 genomic regions. But what I really want to focus on in the next few slides is how do we differentiate between the type of effects that I was talking about. And so this is an example of a direct effect on carcass quality. So what you have here on the left, I have a genomic region on chromosome 26, which has a significant effect on carcass quality. So this specific genomic region is significantly associated with carcass quality. If I look at the genome-wide association study for meat quality at exactly the same position, I do not see an effect. And when I look at meat quality correcting for carcass quality, meaning I'm just including the carcass quality latent variable in the model as a fixed effect, again, I do not see an effect at this particular position. So what this says, it says that what I have here on chromosome 26 only has a direct effect on carcass quality. On the other hand, on chromosome 1, we see an effect from this particular genomic region on carcass quality. I see exactly the same region having an effect on meat quality, and I have exactly the same signal when I correct the meat quality for the carcass quality. And so this is a perfect example of one of those pleiotropic effects that I was talking about, where I know that this particular region on chromosome 1 has a direct effect on both carcass and meat quality. The fact that when I correct for carcass quality, I still see an effect, it means that this is not something which is coming indirectly from the carcass quality. And I'll show you, of course, one of those indirect effects. In this case, chromosome 2, I've got a direct effect on carcass quality. I see the effect when I look at meat quality, but when I correct the meat quality for carcass quality, this signal goes away. So this is indicating that this particular genomic region that I saw in the middle graph on meat quality was really just an indirect effect coming from the carcass quality. And so that just gives us an opportunity to differentiate and identify these pleiotropic effects, which are very important for us. In the end, at the end of the structural equation modeling, we actually ended up with 31 regions that we are focusing on right now. And all those regions are actually related to just 3 overall general mechanisms. One of them is postmortem proteolysis, probably very important, right, for meat quality. But also we have genes related to intramuscular fat composition and deposition, but also cell differentiation and proliferation. And I'm going to move to one more level, and this is the RNA level. So what we wanted to do here was look at the gene expression and combine the gene expression with the genotypic information in order to understand a little bit better the genetic architecture of the meat quality traits. The way we conducted the gene expression experiment, we actually calculated a meat quality index. So we ran a principal component analysis on a number of traits, including marbling, Warner-Brasler shear force, cooking loss, juiciness, tenderness, and connective tissue. And then we used the first 3 principal components to calculate this meat quality index. We wanted to do that because we wanted to select, we didn't have enough money to, of course, do the gene expression on all the animals available, but we wanted to select extreme animals. We selected 80 animals from the extreme distribution of the meat quality index. And so what we ended up with was 40 animals with a low meat quality index. These are animals that had tougher steaks, drier steaks, more connective tissue and less marbling, and 40 animals with a high meat quality index, meaning their steaks were more tender, more juicy, they had less connective tissue, and they also had more marbling. And these are the animals on which we looked at global gene expression using RNA sequencing. So we looked at about 8,800 genes. So these are genes which are expressed in muscle. And what you see here in this graph is it's similar to a Manhattan plot, but you do not have SNP association. You actually have the significance for each gene expression. And so we actually identified 208 genes which were differentially expressed. And I wanted to show you only one of the most significant ones. This is the one right here, ARH-GAP10. This is a gene whose expression was significant when we look at in relation with the meat quality index. And if you look at this graph where it shows you the gene expression level across our 80 animals that we ended up with, you can see that a higher gene expression is related with the lower meat quality index. So at the left end of the graph, you have individuals with high expression of the gene but low meat quality index. And as the expression of the gene goes down, the meat quality index increases. When we look at the function of the gene, I think it's really interesting and it makes a lot of sense. This is a gene which is regulating the actin cytoskeleton remodeling. And so it makes sense from a meat quality standpoint of view that an increase in the expression of this gene makes for a more stable actin cytoskeleton structure, which means a lower meat quality index in these individuals. And so the gene expression data allows us to really get a feel for how are these genes, which are the most important genes related to the meat quality phenotype, and what exactly is their role that they are playing in that particular phenotype. But we wanted to take this one step farther and associated the gene expression with the genotypic information we had available. And so we used the gene expression, so the expression of each single gene as a phenotypic trait, and for each one of them we ran a genome-wide association study using our 250K SNP genotypes. So we had about 8,588 genes, again, these are only genes which are expressed in muscle. For each one of them, we had an expression level. We used that as a trait, and we performed a GWAS. And we identified from this type of analysis, what you identify are not regular or traditional QTLs, but they are expression QTLs. You get a plot like this with a lot of points, but really what you see in that plot on the diagonal is what we call cis expression QTLs. These are QTLs, these are positions in the genome where I have a particular genetic marker, so that would be a SNP in gene A, which is controlling or driving the expression of that particular gene. So that's what we call a cis expression QTL. And those are all the ones on the diagonal. You can also see that most of the expression QTLs that we identified are not on the diagonal. Those would be what we call trans expression QTLs. So that's the situation where you have a particular SNP or a particular marker in gene A, but that SNP is controlling or is driving the expression of a different gene, which is located somewhere else in the genome. So these are trans eQTLs. And so this type of an experiment where we combine gene expression and genome-wide association allows us to identify both types, cis and trans expression QTLs. But probably more importantly, allows us to also identify what we call master regulators. So at some places in the genome, we see these vertical blue lines. Those are spots where we have, we call them hot spots, and that's a SNP or a genetic marker that has at least 20 trans association, means at this position I have a SNP which is associated with differential expression of at least 20 genes. It could be even more, but it needs at least 20 genes to be called a hotspot. And so if that SNP happens in a gene or the gene closest to that particular SNP, we call that a master regulators. Those are the most important ones for us because it means that particular marker is driving the expression of a multitude of genes throughout the genome. From our experiment, we were able to identify 27 of those master regulators. These 27 master regulators, and you have a nice picture of all 27 there, but they are associated with, or they are regulating the expression of 674 genes. And they are really, most of them are structural proteins which are involved in cell signaling, which again makes sense to be related to the mid-quality phenotype that we are interested in. And then what we also wanted to do at the end was say, okay, we used all these different approaches, genome-wide association study for each individual phenotype. We also looked at the latent variables. We looked at the gene expression. Can we identify a number of genes that maybe are present or are significant in more than one approach that we took? And I just wanted to show you just a small part of the table that we put together. So these are all the different type of analysis that we used on the mid-quality traits, and these are just the number of genes. You can see the number of common analyses where they were significant. So we have genes which appear at least 3 or 4 or even 5 different times in different approaches, but that gives us some confidence that those genes are really important in the traits that we are interested in. And so in conclusion, I just wanted to show you how we deal with this complexity level in the mid-quality phenotype, and how do we also combine different layers of omics information. So at the phenotypic level, I think the structural equation modeling really allowed us to simplify things a little bit and reduce the data dimensionality that we are dealing with. At the DNA level, we were able to do the genome-wide association studies on individual traits, but also on the latent variables, identify those pleiotropic effects, and then combining or looking at the gene expression level in samples with extreme meat quality really allowed us to get a better feel for the regulatory network which is related to meat quality in these animals. Of course, the overall goal, or we feel that all these type of analysis are getting us closer to identify, or they are putting us in a position to identify those functional variants which are related to meat quality. I showed you we were able to identify at least 27 master regulators. Those are really the focus of our present research. But we feel that all of these type of approaches are taking us a little bit closer to our overall goal, which is develop genomic tools to select for superior meat quality in our both indicus influenced populations. And I also wanted to give you, before I acknowledge a few people, just a few words about what we are doing right now. So our present focus, as I said, is on these master regulators. How do they affect meat quality, and also how can they be manipulated in the sense of, can we use them to design those genomic tools for our populations? We are also reanalyzing our genome-wide association data using the breed of origin effect. We are doing this right now in our group. In collaboration with the Iowa State University group, which is led by Jim Reese, we are taking a look back at our large Angus data set where they were able to have the whole genome sequence data. So we are going to take a look back at the meat quality and the whole genome sequence in that population. And then in collaboration with the Brazilian group, we would like to combine our population, which is Brahmin influenced, with their population, which is mostly Nellore or Nellore influenced population, and either compare and contrast and see what signals are in common between these both indicus influenced populations and what is different. And hopefully in the near future, we are going to have at least a high density 880K but hopefully whole genome sequence on some of our multibred and Brahmin individuals at the University of Florida. And with that, there are a lot of people to acknowledge, a lot of faculty here at the University of Florida that are involved in this project and are helping with the meat quality part, and also the genomic analysis. But also different institutions that support with the financial aspect of this research. And with that, I'm going to close it here, and I'm going to hand it back to Dar.
Thank you, Raluca, very much. Very, very interesting talk. So you have to unshare it now. There you go. All right, and I'm going to put up my slide that I was supposed to put up, showing another example of you interacting with a nice Bos indicus animal. So and while we're in the transition part, I haven't seen any questions pop up on the chat yet, so I'm going to ask one, and I tell my students all the time, Raluca, that there's no such thing as a dumb question, but we all know that there is. But this may be a dumb question, but I'm going to go ahead and ask it anyway. You showed that the genotypes, that they did differently when we were looking at individual markers, that it was different between how the Brahmin and the Angus cattle performed. Would you expect, or is that a possibility with the master regulators as well, or how is that going to work? If you have these master regulators, are they going to work the same way across the species, or do you know that?
No, that's a great question, and you're right. There are no dumb questions, but this is a great question. I did not look at that yet, and it would be interesting to see. We would expect, I mean, our goal would be to find those functional variants. And if you think of, if we are able to find functional variants which are related to those traits, we would expect the majority of them to be universal, so to work the same way across all the different breeds. I think that what we are seeing is that when we do not have the functional variant, when we just have a marker which is close by, a functional variant, those are different. So those, they have a different face in different breeds. I have to say I did not look at the master regulators which we found, and that would be a great thing, and I'm going to do it the minute we are done here. But I would expect that if we are able to find the functional variants, those they should work the same way. They should have the same face across the different breeds.
Okay, that's kind of what I suspected, but just wanted to make sure. If anyone else has any questions, you can please put it in the chat, and we'll be happy to answer. I think Raluca is going to be able to stay with us to the end in case someone has a question. You can go ahead and type that in. But in the meantime, we're going to go on to our next presenter, and we have Dr. Matt Hersom, also from the University of Florida, that is going to bring us up to date on next year's Beef Improvement Federation meeting that's going to be in Florida. And so with that, Matt, I am going to turn it over to you. If I can figure out how. There we go. It should be in your court. It's up and running.
Okay, great.
So you probably need to put it in presentation mode, Matt.
Yeah,
other than that, it's good to go.
There we go. All right, is that looking the way it should?
Absolutely. Well, no, you're showing us the other screen. You're showing us you might need to move it over to the.
All right, let's try this.
You may have to click on your share button again. Yeah, because it's doing the same. It's your other screen that you need to try and get it to share if you can. Try stop sharing and then try share again, and when it gives you those options, I think you picked the other. If you have a dual screen computer.
So let's see if it will do it this way. Is that right?
Well, you got to share again because it's now. You have to click the share button there at the bottom.
Okay, got it.
There we go. All right. There you go. That's
all right, we're on the right screen this time?
Absolutely.
Okay. All right. Well, good afternoon, everybody. I'm happy to be able to join you this afternoon to talk about the 2020 BIF meeting. And as you can see from this first slide, we are in Florida in 2020 for, I believe, the 52nd annual Beef Improvement Federation. So I've got just a few slides. A lot of times people wonder why Florida, when traditionally the BIF meeting is more in the central part of the country, but certainly from a cattle standpoint, Florida, we've got a large number of cattle in the state of Florida, both beef cattle and dairy cattle. In fact, Florida is, depending on the year, ranks anywhere between 12 and 13 in terms of number of beef cows, and certainly 18th overall. We like to brag that we've got more ranches of 500 cows or more than any other state in the country. And as you look at Beef Magazine, 9 of the top 25 beef cow-calf operations are located in Florida. So we number just under 1 million beef cows, and we will sell and ship out of the state just under 800,000 calves on an annual basis. And again, depending on the calf market, amounting over $500 million in cash receipts for the beef cattle industry in Florida. So some nuts and bolts about the meeting. The BIF meeting is scheduled to start on June 9th, 2020 and run through Friday in June. We've hopefully conveniently planned it to be a destination for everybody to come this year. Certainly, Florida offers a lot of amenities for everybody, and we are hoping that certainly it'll be after school gets out for the summer for most everybody. And from a logistics standpoint, and particularly for our industry people, we are a full week prior to the Florida Cattlemen's Association, so you won't have to pick and choose which one you want to go to. You'd have the opportunity to go to both. Our June timing allows everybody coming in from out of state to get some of the best room rates that are available at hotels in Florida, and will be ahead of the major travel and vacation time for people from coming out of state into Florida. And for those of you that actually care, it is before the official start of the hurricane season. So while I make no guarantees that we can't see a hurricane, the likelihood is extremely small. Our site, and we'll get to that in just a minute, it's going to be convenient for a lot of things. So if you want to come in ahead of time or stay after the meeting, there'll be opportunities we'll be close to the Disney theme parks, Disney Springs, which is a free entry for entertainment and shopping. We're convenient to Universal Studios, the Universal City Walk, again, an entertainment and shopping area, water parks, outlet malls, golf, Gatorland. We're accessible to the Atlantic beaches and Kennedy Space Center. And so hopefully there'll be, aside from the great meeting that we're going to put on, opportunity to have a good vacation along with it. Our meeting site is selected. We'll be at an Embassy Suites, and you can see that is the Embassy Suites, Lake Buena Vista South. And Orlando's big enough, you've got to be specific about which hotel that you're actually after. And so there's the address, Kings Heath, and for whatever reason, Kings is spelled with a Y. In Kissimmee, but Kissimmee and Orlando run together, so it's almost seamless. You don't know which city you're actually in, but that's actually a picture of the hotel there. The Embassy Suites, certainly we're going to have more than adequate meeting space. We'll be self-contained, both with rooms and the conference center attached to the hotel itself. And so just a couple of pictures there of what the hotel looks like. I have attended a number of conferences in this hotel, and it should be more than adequate for our needs. And so we're excited about this hotel space itself. And so, again, we will have run of the house from a room night standpoint, save a few rooms that they reserve for flight crews. But certainly, again, we have more than adequate meeting space for all of our needs to conduct the meeting. This is a map of the generalized area, so I've dropped the pin there at the Embassy Suites, and you can see we're 6 miles from downtown Disney, about a 13-mile mapped or 13-minute mapped transit time, 10 miles to the Magic Kingdom, 14 miles to Universal, 7 miles to SeaWorld, just under 20 miles from the airport, and get to Cocoa Beach and Kennedy Space Center. Depending on that, that's 60 to 70 miles and just right at an hour travel time. So certainly there are transportation options available going to and from Orlando Airport, rental cars, shuttle services, and the like that would all be available or Uber in some cases that would be available for people coming and going. We've got the program pretty well lined out. We're in the process of contacting speakers. And so while I know this is a busy slide, this is the agenda as it stands now. We'll start Tuesday with the BIF will have a board meeting, but in the afternoon start our Young Producers Symposium, trying to look at some topics that would be of interest to young producers. And having been to BIF meetings before, I know that it's just not young people in the audience for that Young Producers Symposium. And so hopefully the topics will be useful to any age a producer. Neogen has contacted us and is interested in having another symposium this year. I believe they alternate years. Again, then Wednesday morning we'll kick off, and we're going to give you a little taste of the history and progress of Gulf Coast cattle enterprises. So certainly people coming from outside the Gulf Coast will give you an idea what goes on down here. We promise this will not be a Brahmin-only meeting, and so, but we do have to show you a few floppy ears and a hump on occasion, and that'll be our opportunity. So we'll look at some of what bull buyers are interested in, some consumer market research and how that relates to the genetic selection that we're all interested in, and then some perspectives from the end users, what they need and what they want. And again, how that relates to the genetics, genetic selection, genetic decisions and tools that we are interested in developing and utilizing. That afternoon then goes to our breakout sessions that are determined by the BIF subcommittees, emerging technology, end product improvement, and selection decisions, and then wrap up that evening with dinner and try and give you a taste of Florida. Hopefully some unique things that you don't normally get to see on a daily basis at home. Thursday, then we've got the idea of integrating sustainability and genetics. What are the trade-offs that we're looking at? And so talk about some of the myths and realities of sustainability, the genetic components of sustainability, and then some animal aspects of sustainability, how genetics integrates there, talking about cow longevity, thermotolerance, milk production, and feed efficiency. Then in the afternoon, the remainder of the breakout sessions, advancements in efficiency and adaptability, genomics and genetic prediction, and then producer applications. Friday, June 12th, we reserved for our tours, and hopefully we've got 2 destinations for our tours that will offer you a great look at what Florida cattle production, ranching looks like, particularly from a large scale standpoint. Our first stop will be Deseret Ranch. Deseret, as you know, is a multi-location holding. One of their largest ranches is here in Florida, so we'll have opportunities to talk about their cattle operations, but certainly they're diversified into citrus, sod, hunting leases, natural resource protections, and so it'll be an excellent stop to look at one of the leading ranches here in the state of Florida. We'll have lunch and then transition to the Kempfer Ranch. The Kempfer Ranch raises purebred Brahmins, Angus cattle, and again, it's a family-owned ranch, actually adjacent to the Deseret Ranch, but certainly offers another unique aspect and look into ranching in Florida and a ranch that's in the seed stock business and also diversified. And then we'll wrap the day up and return to the hotel. So I've got a website listed there. Right now it's not populated with a lot of information, but that will be the stopping point to come back to either between the BIF websites or our University of Florida website to get more information regarding program registration and hotel accessibility. The hotel block is not opened up yet, but we'll probably open it up soon. So check back to see when that becomes available. But certainly I would invite everybody on the webinar. If you're interested, come visit the Sunshine State where the beef industry began. We'd be glad to have you come down. Certainly we are excited and looking forward to putting together a great program that hopefully a little different than what you might get in the upper Midwest, but certainly is very relevant to the entire nation in regards to beef cattle production and genetics. So with that, Dar, if there are questions, I'd be happy to answer them. Raluca is also on the planning committee for the 2020 BIF meeting, and so either one of us could tackle questions about the meeting.
All right, Matt, thank you very much. and we will, if you can stop sharing, we'll move over to the close. But Raluca, we do have a couple of questions for you, so you might want to unmute if you would. The first question we have from, I'm not going to pronounce this correctly, but Dr. Rafiq, I think it is, from Pakistan actually, and he's asking, how good is Warner-Brasler Shear Force for checking meat tenderness?
Yes, that's a great question, and I did not spend a lot of time talking about it, but Warner-Brasler Shear Force, it's actually the most objective measure we have of tenderness. So the challenge with measuring tenderness is not very easy to do. So it's not something like marbling that we can do it in the slaughterhouse right away. So it is very expensive. You need to collect the steak. You need to bring in the steak, cook it, and then you need to do the Warner-Brasler Shear Force. So that's one challenge with selecting for this type of trade, because it's quite expensive and not easy to measure. But the Warner-Brasler Shear Force is the most objective measure that we have, and so very good at predicting tenderness.
And we have a follow-up to that one actually, and I don't think it was intended. This one is coming from Justin Buchanan, and he asked, are there any new tools under development to help the industry collect tenderness phenotypes in the packing plant on a large scale?
That's a great question, and actually Justin was my PhD student when I was at Oklahoma State, so happy to see him in this brown bagger. I do not think, I was just reading the other day, and I do not remember where, but I think there is some technology coming up where they were saying you should be able to do it if it works the way they are planning it. You should be able to do it in the slaughterhouse, and it will take a small sample of the muscle, and it will cook it, and it will be able to measure tenderness. I think that would be a great technology to have. I do not think we have it today, but if that's something in the works, that would be fantastic. So maybe there is something coming up soon.
And he says, thank you. So that's good. That would be great. I guess that is the big trouble with particularly the tenderness trait is just getting the phenotypes for that. It's such an important trait to the industry, but as John Pollock once said, we've kind of got all the low-hanging fruit, and trying to get to this higher stuff is getting, we find the snags then, unfortunately.
Absolutely.
So let me see. I think we may have, nope, I don't see any other questions right now for either of you. Actually, Matt, I had a question, and you may not know the answer, or else it would have probably been up there. Do we have an idea of what registration cost or hotel costs are going to be for BIF?
Hotel costs, I'm trying to recall off the top of my head. With that, it's set already. I want to say, without standing up and walking across my office, I believe it's about $139 a night for a hotel room. I'm pulling up the contract real quick just to look.
All right.
If I can find it fast enough. Not in that folder. I believe that's the ballpark of where we're looking at it is $139. Registration, we have not set that number yet, but at least in our discussions early on, we were under the impression that we will be right in line with what past meetings have been at, and
okay.
So I would not expect I wouldn't expect a huge departure from that registration price.
And I think that anyone that ever attends realizes that the value is extremely good at any venue, and like I say, there's a lot of efforts to keep registration costs under control. So the value for what you get out of BIF is certainly going to be there, I think.
Let me correct myself. The room rate is $149 a night, but certainly, with Embassy Suites, that does come with the free breakfast also. So at least when I travel, I know what the university will reimburse me for breakfast, and it's never enough.
All right. Yeah, that's exactly right. So actually, Raluca, I have one more question for you from Dr. Rafiq. He's asking, is there any effort to investigate the opposite effects of that particular SNP in Taurine and Zebu cattle?
I'm not sure I understand the question, but I think if he's referring to the slide that I showed with that particular marker that had a different effect, I mean, that's a perfect example of marker which is not functional. So that's not the one which is driving the phenotypic variation. It's just close to something which is driving that phenotypic variation. So we see an effect in both breeds, but the beneficial allele is different. So it has exactly the same effect, the same difference in Warner Brotzer shear force between the two groups. It's just a question of which one do you call tough and which one you call tender.
Okay. I think he said he was good with that. So, all right, and we're at our 2 o'clock mark in my time, and at different times for the rest of you. But so we want to close down. I want to thank both Matt and Raluca for great presentations. We really appreciate the information and really appreciate Florida hosting BIF next year. I'm certainly hoping in that taste of Florida that we get some swamp cabbage. It's been a little while since I've had some, so be sure and get that on the menu if you would. But in general, we want to thank everybody and for participating in the Brown Bagger this year. And Dr. Weber will be sending you guys some information, some surveys, if you wouldn't mind filling out. It really does help us out. We seriously do go through those and look at your suggestions. So if you have any suggestions for topics for next year, that would be great. And you can also, as I say, and after some period of time, you can go and find any of these archived presentations that you might be interested in, and they're on the NBCEC website. So thanks again, everybody. And as long as the interest is good, and we had great participation this year, we'll continue to put on these brown baggers every October. So look forward to seeing you all again in Florida and then on the brown bagger next October. Thanks for joining us. Thanks again, guys.
Thanks, Dar.